in my time. So, Mohammed, so the problem with the parking, so just accept that the kids are gonna be late today, some of them, okay, to get started, and then worry about the ones who come in, like they might be as late as not. Okay, all right, all good?
Yeah, uploading an issue I find that is essentially when I sometimes connect to my laptop, it just doesn't turn out well, so I have to always do it manually.
What do you mean when you connect? Well, you've got it set at the PC. You can't use a laptop and get it to go on the PC.
You can't like share display.
No, you cannot. No.
Okay.
So you're either on a laptop or you're on the PC.
Okay.
Okay?
Okay. Thank you.
No problem.
وعليكم ورحمة الله وبركاته everyone. رب اشرح لي صدري ويسر لي أمري واحلل عقدة من لساني يفقهوا قولي.
15 slides to get ready. Get oil and then sort it down. Given how much your microscopes are being used, I would get some 95% on a Kim wipe the bottom of your objectives and also wipe your items, the items. So they're not dirty. And when course of time they get dirty. It's 70. Not the 70, it's the 95. Okay, are we on? I ever put like, something you take your makeup off, you dip it, you wet it, and you wipe it. That's people out later. The color on the last paper, you do something very, very. It's and I would use it your objectives or your objectives. Yeah, you have to go back and read it. Oh, God.
Yeah, you see the one. It's not before.
It's been like 2 hours. It's like he's covered. I'm good for this, and I did present. That you literally say, and I did it, you just think something, what do you want to do? And I was like, we want to write on the back of it. Yeah, writing it.
Hope you're all doing well. So before we move further, hold on. What are the buttons that are on right now? Is anyone turn on? Okay, so if anyone's microscopes are not, like if you have a look now, if you turn on your microscopes and you find that it's too blurry for whatever reason, this is your time to shut it off and then dab a bit of 95% alcohol. You could use it from, I would actually use from the canister or container here, over here, 95% ethanol. It's labeled as such. The one that's 70% is the one on the right hand side from this side, and that's on the other side. Just bear in mind, everyone, this is our final lab for the lab assignment. So I want to make sure that you're all paying attention and making sure that you're able to complete as much as you can to be successful. Any major questions from the lab manual or any questions in general for the lab manual? Let's see. I'm going to just go through a few things to make it easier for everyone to comprehend what they need to submit by the end of today. First off, if you don't have the flow chart for page 33 and don't have the data sheet for page 34, my recommendation is you can continue using them from your lab manual, but I actually need them physically removed so that way I can mark them afterwards. It's going to be the final page or slide for today's lab, but in the meantime, In the meantime, I'll show you what we need to complete first, and that's the spread plating technique. So you probably saw exercise 2, aseptic technique. We've done it in the past, right? As we're aware, using the tubes and being mindful of the Bunsen burner, how we flame our inoculating loops, etc. But in terms of using plates, and this is where everyone will likely come to that side, behind essentially this bench, where right to the right of the incubator, we have different test tubes that will be useful for exercise 3. But just to bear in mind things from the Appendix A that you can have a read of in your lab manual. It involves essentially being mindful, like you would for your bacterium with your test tubes, to be similarly mindful that you're, cleaning up your bench, also making sure that the bacterial agar plate is nearby the Bunsen burner when it's on, not too close, but nearby, so that way it reduces the risk of having bacterium coming from the airborne environment to seep through your agar while they dry. So just bear that in mind, everyone. And of course, similar to using a inoculating loop, we'll be using L rods to spread a bacterium on our plates from the spreading plate technique or spread plating technique. But bear in mind that you, once again, sterilize your instruments before and after touching bacterium. Any questions on that? No? No? Okay. So objectives for today, for the first 15 minutes, I'm going to put a timer as well. Or if you want to look back to keep track of time, we'll put it as 9:42. And actually, at the end of this slide at least, is that you want to be able to, after exercise 3, go straight to exercise 1, which is pretty much completion of the remaining stains you have left for today's practical. We'll be doing the endospore stain first for many of you. We have essentially the fume hood at the very back on my right. So just bear in mind that you're paying attention, especially with this technique, because you're going to be wearing your gloves and you also need your safety glasses to make sure that you don't get in contact. No gloves? Okay. So not as what the lab manual says, but you won't need gloves for today. That was good, but just bear in mind that you're touching like a heating plate, so a hot plate. So just bear in mind that you don't want to burn yourself. In the past, we used to, if I'm not mistaken, but you also nonetheless want to be mindful as you're touching the surface, or as your hand is near the surface of the plate and of the water beaker, that you're not too that you don't burn yourself. So just bear that in mind. As well, if you haven't shown me your hanging drop from last week or your capsule stage, this will be the week for you to do it. But before we move further, I'm just going to go straight to exercise 3 and give you a bit of a demo. So if everyone could come to this side, that would be great. Your bench, you can come or just stand your face. This is our motility test view. Yes. So I'll just put some there because we don't need that right now, but this is where you're going to be focusing for your every Sunday. You're going to be looking back in the past, your test view from last week, and comparing them with the positives, so your positive controls, and negative controls here. Yours are the ones that you see here. And so when you find yours with your ID and your initials, you should be able to compare and note the result in your balance sheet for your observation. And we'll conclude on the right column. That's a separate point, but if there's questions on this, that's as well. Now, for exercise 3, you're working on groups of 4. And you can also all come closer. The idea is that once again, the septic technique involves you being mindful of the instruments you're using and bearing in mind that you don't cross-contaminate any of the samples, nor contaminate them at all. And so the steps involve the following. When you are making use of the So bear in mind, I've touched 70% ethanol. You want to bear in mind that as you turn on the gas, you definitely don't want to have multiple self-help. So let's say I'm going to start. Normally, you want to bear in mind also our air instruments are not too close in contact. You iterate this once again. This is very low, but the point is that you're cleaning up your bench top before you even begin to top. Very important, everyone, that you want to make sure that once again, you don't run the risk of contaminating any of your samples. Cross contamination is where you have multiple bacteria species and one of them gets contaminated by us. So just bear that in mind. Then you can begin the step by essentially being on. Something very generally is a bit more difficult than many of yours, but if you have problems, once again, the blue cone in the middle is what I'm spotted, so just bear that in mind when you have to use let's say, as you're aware from your lab manual, there's different instruments you're using. I brought that one here. You may have, let's say, a set of 3 agar plates as a group. And I'll just show you the instruments that you need for now. You'll be bearing in mind labeling the different canisters or fluids that you have with you. You have 3 for each direction. And pretty much in this one now, the idea is, and you also be labeling with your code number, the dilution, practical, candidates, to confirm the results. Have a follow-up. You also have these pipette holders, which are essentially let's say I'm preparing this setup. And by the way, you guys can all come here as well. I don't want you to miss out. But if you can't see anything, if you can, that's good. So let's say I'm starting this assignment exercise 3. First off, make sure 100 or 200 of your group labels everything so that way it's accurate and you don't have to run the risk of going back and forth and moving around and giggling the cubes and labeling it accordingly. Also very important is that because we're dealing with liquid broth, we suspend your bacteria. You may notice that the pellets at the bottom. So when you're doing dilution, you want to make sure you have all the bacteria suspended. If you don't, the idea is that you're going to have a very minimal amount of bacteria present and growing on your agar. So you definitely want to make sure you resuspend. Let me just show you. This is how it looks before. Right? It's very, if you look very close to the very bottom, you'll notice that the bacteria is highly saturated here because it's forms the color. And then as you either vortex, you don't have a vortex here, but as you essentially keep tapping, you'll notice the bacteria moves up a bit and it's moving, slowly moving. Now it's up. Now you probably see a change in color to a degree. And then you keep stirring or shaking your tube until you have it fully suspended. And this is a good example of the different. Bear that in mind. See how it's different in size or in turbidity. That's like your absorbance reading for the different bacteria. So just bear that in mind, everyone. Very important that you understand accordingly. And just for context, you're using some groups. Any questions regarding that? Now you don't necessarily have to perform the aseptic technique, but once you're ready to perform this experiment, you're going to first take out foods from here. Move on. plate for autoclave is usually somewhere in here. Questions? So now that you're about or in the midst to take out these pipettes, which is going to help you essentially take out 1 mil of bacterium for each inoculant and then resuspend accordingly for each tube. When you do this, this is kind of like your lid for the tubes that we've done in the past. So the neck essentially, your other tube is actually when you're going to replay here. So you're going to replay just like what I'm doing right now. You want to bear in mind that you perform the separate technique of the pipettes that you kind of use at the end. When you're ready, essentially, once it is standing, and you should hope that you expect that you've sterilized your bench accordingly. Then you are essentially taking up with this pipette holder to aspirate in and out your bacteria. Now, I want you to bear in mind that technically speaking, so both that I'm doing now, but technically speaking, because they've already moved up, I wouldn't want to start thinking about you know why? You know why? And going up and down. Your bubbles, and actually, you can actually have bacteria that's easier. Even though essentially you have sterile lines while here, is that here, you're going back here to come in and out. So again, is that if I were to open this, and then essentially understand what it is. Let's see what it does. Place it here in the box. Take it out, you can aspirate, pull it out, and then place it in. Now, for context, let's say this was like what we've done in the past, and you pull it in, moving upwards, your bacterium. When it moves up, the volume moves up essentially. So just bear in mind that you're going to 1 mil, then you're adding the appropriate amount of bacterium. And then you flame your lid once again, you repeat the process. And then, accordingly, you're labeled 10 to the power of -1 dilution of glass would be thereafter aspirated or essentially inoculated with 1 mil of your saline, pretty much salt with water. Now, the other thing is, once you've done that step, you're going to be adding in the appropriate amount into your your agar plates. Bear in mind that's 0.1 mil, so that influences the dilution. You're going down another power. So if there was negative 10 to the -1 here, and you're adding 0.1 mils here, and it's going to be 10 to the -2. Just bear that in mind, and the process repeats itself as you take in the following, essentially, once you essentially resuspend, right, the saline and dilution, you're going to repeat the process, aseptic technique. You can use either the same or separate methods for each step, and then essentially take it on to the following two. I prefer to essentially keep the same method. You don't have to use multiple ones, but if you find that you cross-contaminated, that's where you want to be mindful. And essentially, you repeat the process back and forth across the tubes, and then at the very end, you essentially can take 0.1 mils of each diluted, spread it across with the elbow rod, and then you should be able to place it back taped or labeled and return it to me so we can place it in any such. So the reason I said you don't have to essentially clean to a degree is because you're going to be sterilizing students at the very beginning, all of them, at the transit essentially as we move on from one tube to the next. Technically speaking, within this set of only 3 tubes or 3 sterile tubes, you can simply use the same method. So you don't have to worry about sterilization. When you're done, you essentially place it here. spreading from 0.1 mils. When you reach that step, that's where you want to bear in mind you converted your agars, you're performing the aseptic technique because essentially you would have had 95% ethanol at the food, which is very not possible, but the one that's right behind. And essentially that's 95%. You would have added it to the Petri dish. You use your L-rod, you use flame your L-rod, to fill your, the pot. We'll take the one. Essentially, the flavor L-rod before you even touch the bacteria. Bear in mind, you first put 95% ethanol, then you flame. Like you whisk it a bit, then you flame. But also bear in mind that not flammable, so you don't want to burn. You don't want to catch on fire. So just let it heat up if you're not really blue. When you reach that step, we'll try to reach that soon. But then just bear in mind that you do this process, you can either use the turntable, you can like pottery, and then essentially distributing evenly the bacteria. And when you reach that stage or near that end of that stage, the 0.1 mils added here into your essentially agar tube, then you let me agar plate, then you let me know. And then you just repeat that for all of these units. Right? So you have 3 agar tubes, agar plates for the 3 sterile tubes here, which are your dilutants across different degrees of bacterial dilution. That's serial dilution for you all. We have, okay, so let's go straight ahead. Oh, yeah.
In the lab manual, it said that we're doing the serial dilution per bench, and then it said that we're doing spread plating per group. So is it like partnered or?
Both will be in group section.
Okay.
The reason being is because we only have 3 groups or instead of 4 essentially. In total, actually, yeah. So that's actually why we won't have enough to really improve pairs, but it's okay. Essentially, keep the same level of consistency as we do zero dilution and then spread plating. If you want to make it even easier, you can also separate into pairs to do spread plating and serial dilutions. Once the serial dilutions are done, then set up 2 like 5 pair people. One that's labeled everything, one is ready to essentially use the pipette and to inoculate and dilute your tubes. Once that's done, you don't have to also do it in pairs. You can do it all as a group. But the idea is that once that's done, you can either place it in the spread plating, which is what I've talked about before. Essentially, spread plating technique is that you are oscillating your bacteria in different directions, spreading it to make it as evenly distributed as possible, following each technique. And then at the end, once you're done that, across all 3 dilutions that are labeled onto your agar plates, you place them into the incubator, and you're ready to complete that for your group numbers, yes. I can divide them accordingly. In the meantime, I would just say everyone get ready now. Any other questions? Before you begin, make sure that security ethanol, 70% ethanol on your bench. If you want to, literally all have them. Containers of 70% ethanol here, 95% ethanol for your all rods are here. So bear in mind that before you put on your Bunsen burners, the ethanol that you have with you is sufficient for the exercises and that they're not too close. Like this is a good example to just bear in mind that way you don't run the risk of having anything. The motility questions you can use afterwards because those are observations, for observation. And they're relative to Yes. Not yes. Serial dilution is spread plating. So everyone, this is the order of today. We're going to be focusing now on spread plating and serial dilutions. So bear that in mind, everyone. As I mentioned earlier, you need to sterilize your bench. Make sure you have all equipment necessary. You have the pipettes, the pipette holders. You have your agar plates labeled, your tubes labeled, and essentially bear in mind that you have 95% ethanol available and 70% if needed on your benches before you turn on the Bunsen burner. Just bear that in mind so that way you don't run the risk of having limited supply for sterilization, and that way you don't have to repeat the process multiple times. If you want my recommendation, do it as an entire group. That way everyone's able to see and visualize how you do serial dilutions, which is very important for your lab exam, but also importantly, aseptic technique and spread plating. So make sure you have all items before you turn on your Bunsen burner. Make sure as well your Bunsen burner is not too high. And if you have problems turning it on, let me know. In the meantime, I'm going to start separating you into groups now. So just bear in mind your number to help you label your agar plates. So bear this in mind, everyone. On my left, Rahma, your group, the three of you with Christina, three of you, and I forget your first name, but Core. I know I'll call you Miss Core. The three of you are one group, group one. The three of you are group two. The four of you are group three. Now, Rahman, your group four. Your group is four. Joseph Ferraro, the group of you is four. Your group is five, and your group. Just make sure that you label your numbers for any other experiments. Okay. How's everyone doing? I could give you until 10 o'clock to be able to complete this. So hopefully you should be able to complete a good chunk of today with time. Okay, I'm gonna make mention. Give me one moment. Labeling your agar plates, I recommend labeling them on the sides, on the periphery. And I'm gonna show you why. So imagine this is your agar plate. Everyone please pay attention because sometimes this is a difficult process for weeks to come. Imagine this is your agar plate. You want to make sure you're labeling your group as practical, right? PR8, that's 2. And then your group, let's say, is G or group 3. And then your dilution factor, let's say it's 10 to the minus 2. Bear in mind that you're following the lab manual. And you can also put the date, today is I think the 28, okay? So 1-28, you put 2025 or 28. And the idea is that you want to do it on the periphery. Why? Because essentially in the weeks to come, you're going to be counting these bacteria. If you have them all labeled at the very bottom, it's going to be difficult for you to actually count the bacteria, which are only small, right? So ideally, you're going to have bacteria all around. Some of them may overlap, right, with your labels. So it's definitely ideal that you have them essentially labeled on the periphery to avoid having so much overlap when you're completing your spreading. Just bear that in mind. They don't always look round, as we know from before. Some of them are more filamentous. Some are, even though these are more in the microscope, the idea is that you're able to see, hopefully, bacteria without much overlap. So just bear that in mind, everyone. You can label it on the bottom. So agar bottom, I'm going to say agar bottom. Agar bottom is like this. So write down on the bottom of the agar, essentially on the side. So let's say I'm practical 6. Practical 2, group 6. PRA 2, group 6, and the date, and the dilution. So bear that in mind. That's all. Oh, the tube? You can write on the tube itself. If you need to. You can, but the issue is that
In the last class that I was what was the
you don't, because we'll know where it's placed. Group easy. Anyone having problems, difficulty, concerns? I'm putting a timer to make it easier for you to bear in mind of time. So just bear that in mind, everyone. Your group number should suffice.
Do we have the dilution with our practical and our group and our stuff?
Yes, dilution should be. 4 things to label: your practical, the group, the dilution, and the date. Bear that in mind. I'm just going to check a few things. Once converting is also on, and you're having problems bringing it on. So here's a question. How much volume are you adding of your bacterium to start off with? 1 mil. Is 1 mil at the top or at the bottom? At the bottom. So some of you may have pipettes that are inverted to show 9, right? Because you go up from 0 all the way to 10 at the bottom. Bear in mind you're subtracting them. So 1 mil is going to be where you reach 9 on the pipette. And if you have the opposite, then it would just be 1. If you have any other questions, let me know. See it. So just to also clarify, some of you have pipettes that go up to 10 mils, and if you go essentially as I showed earlier there, if it goes to 1, that indicates you have 1 mil out of that 10, right? But if your pipette only goes to 1 mil, then that one increment is actually 0.1. So bear that in mind. You're adding in the right amount. So the one that's labeled as 10 indicates that you're going to be using it for the serial dilution, right? But when you're doing spread plating, you're using 0.1. You can use, if yours only goes up to 1 mil, that's also okay, because all you have to bear in mind is that for serial dilution, you go all the way up to the top where you reach 0. And if you're doing spread plating, sorry, you go to 0.1, which is where the 9 go up to 0 at the increment. Where you see it at 1 mil. If you have a 1 mil pipette, some of you may have 10 mil. If you only have a 1 mil pipette, you go up to 0. Who here has a 10 mil pipette? Raise your hand. This is a 1 mil. Anyone have a 10 mil pipette? Raise your hand. Who has, okay, so if all of you have a 10, if you have a 1 mil pipette, I'm just going to repeat this one more time, because in the past we had 10 and 1 mil, so that's why I wanted to clarify it. Essentially, if you have all of you 1 mil pipettes, serial dilutions which go up to 1 mil, go up to essentially the 0 increment. Once you've done that step, right, in pairs or the entire set of 4, when you're going to spread plating, you're doing only 0.1 mil. And that's when you go up to 0.9 mil as your increment. So essentially up to there. That's 0.1 from the top to the bottom of 1 mil. Okay. No. If you have to repeat that, let's say you added 0.1, you add an additional 0.9.
Keep jamming it down the glass. Good though.
This is and what you see representing or having similar characteristics be a simple rate. Essentially, at the very bottom of the the bacteria are compelling.
We're gonna take you later. I don't know.
Doing so far? I'll add an extra 5 minutes. I want you to be all efficient with timing, so that way, hopefully, you don't run the risk of being ill prepped for completing your lab assignment. So I'm giving you about less than 6 minutes, 5 minutes and 45 seconds to complete this one more round. Please make sure that once you're done, you've labeled them and placed the agar on top for your plates, and that way we can place them into the incubator. Anyone having difficulties? Everyone okay? Make sure you show that serial dilution before or after jumping down with my message. It's okay for you to make use of them for the other serial dilutions because they're from the same bacterium, and you would have definitely cleared the list of those other serial dilutions. So, just so everyone's clear, you could actually put in 95% ethanol and then flame after it's been wet. So bear in mind that you, let's say, you find in how much, either way, invert or place the agar on top. And you're done as well.
Yeah, yeah.
On the slide, please. Or somebody should, let's say, all of the materials we use for today all of them can be placed back to where I took them and actually where they belong. Just place them where you found them earlier, and it should be okay. Yes. You can place the cereal dilutions on a single blue tray. If you find one already there, just place them all together with your tube.
Thank you.
Nope. And then I'll talk a bit more. You can leave it on. Actually, with the percentage, I would recommend right? You can put it to it's possible. If you put meters, then you can just feed it some kind of thing and then if you have people need your car, please let me know now. Oh, sorry. But what I meant by three. For there, if you can give it to me or bring it to the incubator as soon as you can, I'd be really appreciate. So I just want to make sure everyone's also paying attention in time. Great job that you're able to complete this thus far. We're gonna move now to the endospore stain. So, first off, does anyone know what's the function of an endospore in bacteria? Raise your hand. Okay. Yep. Okay, that's good. So desiccation is one of the benefits of an endospore. It prevents bacterium it actually helps them resist certain conditions, harsh conditions like increases in temperature, increases in or decreases in humidity and in dryness or desiccation, and helps protect them from host immunity. So just bear that in mind. Now, you have the endospore stain, you have the motility test, right? That you probably saw right there in the middle. Now, I want to make sure that you're able to complete everything else. You have the endospore capsule. Let's just write them down so everyone can also feel. Endospore, like the equipment is already prepped up. Gram stain, capsule stain, Is anyone noticing anything missing, or is that it? Apart from the hanging drop. Anything else? From your dad, please. So that appears to be it. Those are the main five. But I want us to just bear in mind that we're paying attention because within the time frame of today, we have to complete all five if we haven't done them last week. Motility test tubes should be simple for everyone. You see the results down there. So that should be a check. Now we've done aseptic technique, the test from before. You may be pending on the gram stain and also on the capsule and maybe on the hanging drop. If, and this is what the order I would recommend everyone going with, from first to fifth or last, because the endospore stain is based on the, like essentially one of the limiting reagents you have is the heat bath, the water bath with water. The water is getting evaporated, so there's limited amount of time that you have to complete the endospore stain. So I'm gonna ask, once you're completed or once you've made your smears, your 3 smears of your unknown in the middle, make sure you've labeled your slides once again. Your negative on the left, your positive on the right. Once you're done, once you've heat dried, or sorry, like air dried your smears, that's where, as groups, you're going to be going there to make use of the endospore stains. I want you to bear in mind that because we have limited space for the endospore stain to be done and limited time as well, that if you find more than 4 people there, because at max 4 slides can be placed in both beakers, and just bear in mind that those are beakers, right? So you don't want your slide to fall in. So bear in mind that you're paying close attention. If you find that the endospore stain location or designated area is being made in use, I want you to focus now on the gram and capsule stain. The gram stain you have to submit today. The endospore stain you also have to submit. Your flow chart, right? The flow chart, I mean the one that I showed about the 6 bacteria last week. You have them, let's say all 6 here. You have a given test, let's say gram staining, right? Gram stain. And you have a set that are positive, a set that are negative. 5 are positive, 1 is negative. You separate them accordingly into the different categories, making sure that you mention the category and if it's positive or negative, motile, non-motile, encapsulated, non-encapsulated, with plus the test. So bear that in mind. There's 3 different components to this for each level, like the test needs essentially. And then you return as well your data sheet with your observation, your name, your unknown, and what you believe the unknown is indeed. So just bear that in mind. Fill in all details. If you are able to complete this, even each test hypothetically in a given amount of time, look at the time now, it's about 10:19. You have an hour and a half remaining. 90 minutes to complete, even if you were to begin from the beginning, motility tests you already have. So really, out of the 4 tests remaining, you have about, let's say even 15 minutes or 20, 90 divided by 4 is like 22.5. So that's ample amount of time. And I don't want you to just look at that number. I want you to think how efficient you can be trying to complete this, let's say in 10 minutes each. That way, even if you haven't, let's say, prepared the flow chart or the data sheet, you can write them in a separate piece of paper. You can even cut them out of your lab notebook, and that way I can mark it when you return at the end of today's practical and submit those all 3 to me, essentially. Bunsen burner, okay? Yeah? Yeah? I heard a Bunsen burner go on. Unknowns, your unknowns. The ionized water will be here, so feel like it's right here. That's where you're filling the ionized water. There was a question on that side. What was your question? Okay. Another thing, you have slides as well from last week. You can look at them on the left-hand side. If you find that your gram stain is good, look at the microscope or your capsule stain. You've labeled them. And you're ready to submit them, go to the next steps. Go to the endospore stain, go to the capsule stain if you haven't completed it yet. As well, make sure that you make note of those different results in your observations and the conclusions you can make all together. Somebody,
Yes.
if anyone needs their unknown, motility test tubes, I'll just show it to you once again. Everyone should actually come here if you aren't doing something urgently important, because I wanted to put you for more time. Bear in mind the following: a positive looks like this, a negative looks like this. Reason being is because there's a hole at the end of the small tooth, right? It's in the middle of your larger one. So if the bacteria will move out and you guys see my reaction to that. If your bacteria will move out, you go under the tooth like here and fill up the entire ER looking red, like it's positive control. If they're not out or negative like here, then a simple streak would really pretty much maintain the range only here, not throughout the entire look. So when you're looking at yours, have a look, reflect back when you see multiple streaks, like multiple tags or like marks within that cylinder, then the smaller one, then that's like the normal tile, whereas if the entire group itself, is red, that's an indicator of positive or negative. You can also use that against your yes, that's positive. Some of you yes. Right?
Bacteria beautifully.
Everyone else, your Bunsen burners, are they hot? If, so 2 points. Your question was, oh yeah. If you don't have enough bacterium in your unknown, which actually is, like bear in mind that you should have enough bacterium there for endospore stain, and likewise for the other stains. But if you don't, let me know. So that way, hopefully, we can have a look at your NA time. Also, when you're using the Bunsen burner, you can put your inoculation loop this way. So you could do the other way, like the other way. Yeah, this way. That way it doesn't recruit everything, but perfect. Now the second question is your slide. If you're done with your gram and capsule stain slides, gram and endospore stain slides, I want you to have them labeled and side by side so that way I can mark them easily. So for the meantime, if you're already done your gram stain from last week and you don't need to repeat it, then just keep them with you until you complete the endospore stain. Make sure that by the end of today's lab, you need to have all of your slides for at least the endospore and gram stain back here. Just the endospore and gram stain. The capsule is for just observations to know in your data sheet. But bear in mind that the endospore and the gram stain need to be submitted. And that should be enough. So I'm gonna start asking if people can move towards doing the endospore stain because it is higher priority for today. With the submission of your slides. So just bear in mind that you have 35 minutes to complete this. Go to your endospore stain if you haven't done so already, so that way we can maximize efficiency for today. By the way, there's four slides available for the fume hood right now. So because I've given you 35 minutes, think of it like this way. The fume hood should be used 5 minutes for 4 people together, meaning that if you do the math, with the 24 of us here, we should be done by 30 minutes. So bear in mind, I need to see in the next few minutes people there to be as efficient as possible. Number spew is high, too high. Whoever has it now on too high, please shut it off because you don't want to start a fire. Anyone has it again? Someone's is too high. Please, okay, I'm gonna ask if everyone can shut it off right now. Shut off your Bunsen burner because it's been much more higher than it would have been from the past, and I don't want anyone starting a fire. So just shut it off for the next moment. Everyone shut it off. Everyone shut it off. Everyone shut it off. Because right there, I smell what could have been a fire. So just please be careful and don't turn it on all the way. I know some of your Bunsen burners. Please be careful, especially the ethanol, that you won't start a fire. Okay. If you're having difficulty, let me know, but as always, try to lower it slowly, so you don't run the risk of starting a fire.
Yeah.
Everybody okay? Okay. Anyone coming to the capsule state? Sorry, endoscore state right now. If so, this is your time to come here. I'm just going to help showcase a few points. Please bear in mind, remember that you only have a finite amount of time to do the endoscore state. If you're not completing it in the next 35 minutes, you don't have time to do it in the practical. So bear in mind that you're coming here paying attention. Focus on the endoscore state because it's time sensitive. So perfect. Now, so, let me look at everyone have a look at I'm just going to explain this to make it easier. So she's adding this, she's first using slide. On the surface of the center, if you need more, just add in. I will see one that's a bit more easier to grasp now, and it's the one shown on the right. But the point that you have to bear in mind is that once it air dries, you have to keep adding more. So just if you see it essentially becoming more evaporated, add in a bit more malachite green, every so often, can unresolved findings, mutations, keep adding it once it runs. Essentially, it'll take a bit of time. It's important to note that you have movement. So research are useful to carry in and out these slides because you don't want them to go into the ether. That's a very other one. Um, So usually, if you do have the piece as I would say so in the meantime, definitely recommend everyone slides. And just bear in mind you have the timer there. Yes, yes. Having trouble navigating this, just let me know. If you're also having difficulty holding your slides, also let me know. You have a dream watch, very good. Completing that. Yes.
Think about it. I just do ground statement and.
Yeah.
Oh. Everything okay?
Yeah.
Okay. Make sure you keep a track of time. There is a stopwatch there on the slide, on the right of the slide. Okay, for that. Yeah. Some of the Bunsen burners are a bit sensitive. If you start off high, just make sure to go more down low. Put them like this. And another thing I think is important, if you it's a things because sometimes if it doesn't go on, you swallow, you're not going to this. You know, but that's good because you put it on the so you should do it as well. I would say yes. The honestly, you want to do it very well. I would wait another how long people. So maybe like the last minute. If you do well, the in the sports day, which is nice, it's like you're adding like wrinkles into the cookie or cupcake. If you're adding like if you've done it well, then at the last minute or two, you really kind of add to your job. When you do it, when you do it, it's that then you get all positive. But that's in the sport or MVP for that. And then go to the other level. It just fell off on the yeah. Do you still need it? Like, if you done the exercise, or do you need it for?
Just like. Yeah, so hot. Oh, sorry. Oh. Let's see. You are this. No, not yet.
I just want it won't be done.
Okay.
Because you're focusing on student.
Yeah.
If you can pass it on somebody else, then do it. Joseph, grab it from there. By the way, if your bench is not using a Bunsen burner, please shut it off.
Um. No, the blotting paper, is that the building paper or the Kim white?
For which stuff?
Like blotting,
For
like the dry.
endospores or? Okay. Anyone have mendable tape?
We do at ours.
Does anyone have two?
Ashley, would you go to the syllabus? They have. Oh.
You guys don't want to overdo it. Just keep an eye on time, and when you reach the last minute or so, just see if it's dry. Then you can have it properly. The group was asking for it. Oh, let me see this. How many of you are still waiting to do their endospore tape outside? That here? Okay, 1, 2. So may I ask, what are you guys doing right now? Grand state?
I'm waiting for it to dry and then.
Okay. Okay. So that's good. I just want to make sure though, in the next few minutes, ideally in about 5 minutes, everyone should be on the stage of heat drying or at least completing their endospore stain. So just bear in mind timing for everyone. Let's see.
There's no,
Did you look at it?
I don't think
And then all of my phone photos is watching.
Not moving on to your endospores thing that we know. I mean, to increase the temperature. That way we're able to see identity that we have. I'm sorry, endospores. I want to also mention two things about the endospore, sorry, the capsule stain. I know from the past some of you were having difficulty doing this. Really and generally speaking, most of the observations, if they're randomly, like essentially, if you consider all of the species minus L. lactis on skim milk, all of them generally will be negative. So I know some of you had questions about like if it's positive or negative based on the resolution. Some of them appear positive, but it could be the case that you don't you find essentially a bacterium that isn't L. lactis that covers more of the properties from your results. So just bear in mind that the capsule stain is not a diagnostic test to discriminate or differentiate the results like I showed for the flow chart. However, you're using it as a dot in your diagram and for your observations to note down what you see and what you perceive your bacterium could or could not be, essentially. If there's, I think that should be it. Yes.
I know.
That's my problem.
It's always right. Yeah, always trying
This is recorded.
No, I didn't keep on. If you look at like my name, like seven, all that. You just go to 30 years of work.
Any other questions?
Yes.
Yeah. One moment. There was a question from.
Getting the L out.
So before I answer questions, um, Joseph, is are the beakers heating up? Like, do you still see steam and like, yeah, you guys see steam? Both of them, yeah. Okay, so I have a few questions. Raise of hands if you've done your endospore stain. Raise your hands. Raise your hands if you're done. If you're in the process of doing your endospore stain, or you're done it essentially. Keep them raised. Keep them raised. Okay. How many of you have not done your endospore stain as of yet? Like, aren't like on the stage of at least putting malachite green? Raise your hand as well for this one. Okay. Then okay. So generally speaking, everyone has done the endospore stain. Am I right? Okay. No? Okay. If you're in the process of doing it, and have already heat dried or air dried your slides, then what we'll do is give you an extra 10 minutes. But I want to make sure that this will be the final time. Okay. Let's see. Oh, we have so less than an hour. Let's see. So I'm just going to put some time. We have about 53 minutes. 3 minutes for cleanup. So we'll say 50 minutes left. How many of you are not done their gram stain and want to redo it now? Please raise your hands. Who is finishing off a second gram stain experiment? Like who's doing it again? Okay, raise your hand. Everyone, please raise your hand. Okay. Okay. And who has to do their capsule stain?
No.
Now who has to do their capsule stain? Okay. So let's have a look now. Motility test should be done. Hangings are up. If I give you guys, let's see. Motility capsule.
What's more important for us to do, the hanging drop or the gram stain?
Okay.
Gram stain, okay.
You're in order. So essentially, focus on what you need to complete and submit for today. Endoscore, gram stain, both need to be submitted with the flowchart data sheet. And then the capsule stain, motility test, and hanging drop are for your observations for the data sheet. But I don't need them per se to be returned to me. So just bear in mind that you complete these as well as these for your observations. I'm just bearing in mind time. So we have 23 minutes. I want you to be done. And this is a hard deadline for you all.
15 minutes, 15 minutes.
Yes, but I'm putting 10 here so that way, like if for whatever reason people are like not complete one of the following, then at least they'll have an extra time to go.
Can I take a picture of my endospores
You can.
for my unknown? Can you also look at it?
Yes.
Thank you.
No. Yeah.
But then do you rinse off with water or just add it 5 seconds, dump it out, add it. Just add it and then rinse it.
I didn't rinse with water. Sorry, you've already added the one, right?
Yeah, and then add.
No, I don't. Decolorize and then yeah, you can rinse with water. Or actually, you've added in so 95%, and then your next step is. That's what I was thinking. Yeah. You could use your phones to make note of figures for your endospore stain for the post lab for this week. Just bear in mind that, of course, you don't throw away your endospore stain slide and you've labeled them accurately.
Yes. Just.
Yeah, I did. Was that paper? So- Fucking oil here. Fucking good this, but it's clear. So. And therefore, everybody.
Had a question. Your question about endospores? Yeah. Before we move to Great. You guys have questions? Who else has any questions that maybe I missed? Any questions? Yeah? Okay. If there's any outstanding questions there.
We can see you books on the garbage laws and
Okay, so just want to confirm everyone done. Well, before I say other things, I want to give like a kind note regarding this. I know some of you maybe are having questions with your endospore stain. Many of you may not be seeing your positive control. Believe it or not, this happened the same, like this has happened in the past. I'm not too concerned about the controls, at least the positive, if you don't see an endospore. However, if I do see a positive like result in your negative control, then perhaps that's your time to actually relabel your slide because you definitely want to have the right label for the right smear. So just bear that in mind. And you can also like there was an endospore, there's another one. The hanging drop. I'll give you about now 5 minutes to work on actually, how many of you have not completed yet the endospore stain? Raise your hand. One. How many of you have not done an endospore stain as of yet? Okay. So in the meantime, you have 5 minutes to work on the endospore to complete it, to pretty much go to the bench right there. In the meantime, I want any group or any member who has not done the gram or capsule stain to start working on it now. You can have 7.5 minutes to work on one and then 7.5 on the other. That way you're making max use of your time. So just bear that in mind. You could, air dry them together, the two slides if you need to. If you're done one or the other, the gram or capsule, you can move on to the hanging gel or your motility test here. Uh, just want to mention an important point as well. Many of you have many of you have coccus-shaped bacterium, right? That should actually help you denote and differentiate an amount of them, essentially, from which ones could be yours. Now, bear in mind as well that if you look at the table of observations, page 33, that you would have expected only a rod shape to be subtilis, to be endospore positive. Now, what that means is essentially what we're seeing often the case in our bacteria. And I know many of you have said it's positive, especially your group have said it's positive. And it's okay if it isn't. In fact, many times it'll be perfectly normal. Imagine this is your red shaped endospore from your, bacterium from your unknown. Bear in mind that some of you are just seeing a red circle inside your bacterium. I know for many of you, I would have assumed that it would be positive of having an endospore. Please also pay attention because I know I've repeated this multiple times, but I want to make this as a clarification. If this is your bacterium and it's rod-shaped, you can assume it is endospore negative. Reason being, endospore negative if it's coccus-shaped, rod-shaped, depends. But generally speaking, I haven't seen an endospore from many of you. So just keep that in mind. I know I've said before that it could be positive, but it's a diffraction that makes it appear as a positive, false positive. So just bear in mind, if it's like circle shaped, you can assume it's negative if you haven't seen a green circle inside. And you could write that down for your observation.
Wait. If it's.
I don't know.
But you said that I was positive.
I know. I know I've said it before. And it's not the time to look at updates because you may assume wrong that like you may, because the endospore stage is supposed to be discriminative. It helps you distinguish your test.
So
But I would say go if it's not rod shaped.
then it's
Go,
negative.
yeah. I just want to say, don't worry too much about let's say minute details. I want you to be able to complete the lab with excellency, doing your best, being proficient, right? And being able to at least deduce where your bacterium could possibly be based on 3 or 4 or all 5. Like as many tests as you can. But it's very important that we don't like I want to make sure that you're able to know the majority of the test results that you're trying to conclude appropriately, even from your own. You have to redo one of your unknowns or the controls. That's okay. We do have a bit of time, but that would only be for one extra slide. So bear that in mind. You do it appropriately, your smearing technique, 2 drops of water at max, and then you add in, if it's a solid culture, a fine amount. As once again, the inoculating loop is this size, let's say. Let's say this is the size of your inoculating loop. A fine amount should be enough. Like that amount, and then smear it in accordingly into that liquid. And if it's in just 1 or 2 smears on a slide, just label it accordingly. You could use the endospore stain to repeat the process for your positive control if you're doing it again. But just bear in mind that because of the time we have now, you really have to be able to complete. Ideally. And I want to find out from everyone, has everyone done the endospore stain? Yes? Who has not done the endospore stain? Raise your hand now. Okay. Now I'm going to ask, who is working on their, sorry. gram stain. Who's working on the gram stain? Okay. Gram stain, that's okay. So in the next 6-ish minutes, 6.5 minutes, complete your gram stain. If you already done your gram stain and need to work on your capsule, this is your time to do so. Bear in mind that we have finite amount of time. So after this, if there's any other stains needed to be completed, you have only 5 minutes to do it, which is sufficient, but just bear that in mind. I would recommend if you have both stains to complete, the capsule and gram stain, capsule for your observations, gram to submit, do them both together. Air dry them together, put the same unknowns, the same unknown, put the different controls, or at least the controls that you have with you, the control that you need, and air dry them together. Don't do them separately, so that way you can maximize your timing for today. And then after that, you're going to need to complete the hanging drop in the last 10 minutes. Yes.
On the scale of 40, I said into 100, is that okay? Because I can't see it properly. Yeah, I know. But like, I'm not.
I would recommend if you have gram variable results and you're taking a figure, like a photo of it, you need to show it to me because I don't want you just to rely on your photo essentially. Resolution's better by your eye, and also to denote other locations in your stain is also important, or of your smear. I'll have a look at this. Is that or
No. Yeah.
If you have only your data sheet printed out, or you have your flowchart on your lab notebook, you have two options. I recommend personally redoing it. You still have hopefully time to redo it, and then take the photo of, or send me a photo, or you give me the entirety of them together. The other option is taking it out of your lab notebook. I prefer you not doing this because I want you to keep it all intact. If you have the time, if you've done the different steps I've said thus far, or you're just waiting for the next steps, draw it out quickly on the other side of your sheet. So, were you able to separate all of them? If you're able to separate them, then this will be a problem. However, if you want, you'll have maybe which of the following unlocked from these strings, such as you would have only be secure on one side, and then everything else. Because it's probably after so essentially you can move these two steps. Notice how essentially, like I would say, or you could say, okay. And then you can just like put the two bottom round or something.
And then times it with water, add them for 100, and then I will depolarize with 3 times, right?
And if you complete, this is your final turn to do so. Okay, I'm just adding an extra minute for you guys. This is your final turn, or roughly your final turn to do the final stain, either your capsule or your gram. I know you have some of you have hands raised. In the meantime, because it may take me some time to answer everyone's questions, see any final stains that you need to do for your slides, label them. If you're done, place them into the slide rack on the left-hand side. I'm just gonna ask the team of your question. What's your question?
It was just about how to actually shape up.
I want you to be able to, because the color is something that you need to do. Look around and you see more purple than pink, and that's not.
Well, it's fine, just the actual shape, I'm like unsure.
You have to stop.
It's the broad.
And then have a look and see options on a broad shape. Do it in order. Sorry. You can watch. If you're not extra there, then there is like yours. Okay, then you go back. Yeah. Amon, you had a question, right? Or was someone in your side who had a question? Repeat up or? No. Now we're done actually. So, you want to do something else? Yes, thank you. Yeah. Wait, how do you use
This looks like fun. And see,
if you need, you can just figure out. It's okay. I don't mind. It's okay. That was, but they should take a positive message. You didn't solve it. It is possible that you perfect. But if you have okay, so I know I've answered many questions from this side. I want to finalize. Hopefully, everyone here has been able to finish their slides. This is your time to start labeling them, right? In terms of your unknown, your controls for both endospore and gram stage. For your capsule stain, you can note the observation. And in about a minute or two after these questions, we'll have to go straight ahead to the hanging drop. If you haven't done the hanging drop before, this is your time to look at it. Really spend at max 10 minutes. If not, pretty much use your observations to deduce what your bacterium would be. But generally speaking, just spend the next 10 minutes. I'm not even, I would say yes, 10 minutes, even 7, 8 minutes to just focus on the hanging drop. The hanging drop, essentially after you've done the inversions, go to 100x. Make sure you add it in oil, and then pep at the edge of your droplet, essentially. See if the bacteria are moving in multiple directions, as motility in a directional manner, or if they're just vibrating, that would be motility. Do you have them right beside each other, or?
Yeah, they're right beside me. But then after so they're not truly that it's right.
I'll just make mention of 2 points. Everyone just bear this in mind. For the motility test, you have true motile, non-motile, or red and white, or sorry, or clear, for your tubes, right? Now, for the hanging drop, you're gonna say either true motility or Browninian motility as your conclusions. Just bear that in mind. For observations, true motility is directional, i.e. they move into multiple directions like they're in a race, whereas Browninian, they're vibrating. They're just huddling around or threading around in circles. So just bear that in mind for your hanging drop. Yes.
So what my back drop and I know that there would be I did earlier and no, I did my motility
Yeah. Yes, screaming because it was. Oh. That's the two. Okay, we'll do wrong direction. You see this?
So So remember, everyone, you should have likely been done your hanging drop by now, or in the next 3 minutes. So just bear that in mind. Timing wise, you need to be done in the next 3 minutes, and then following that, the last 3 minutes for today's class will be handing in your slides and your flow charts, and the data. Just bear that in mind. Now, when you're ready to submit, just let me know. You've organized your slides, confirm them with me. If you have more than 1 gram stain slide here, that's also okay. Just let me know. I'll be very honest. I want to make sure that you guys do your best in this practical, and I'll try to take into account the best news that you have. But you just have to make sure that you're clear. You're showing with consistency which traits you find from your slides, so that way you match up with not only your observations and conclusions, but for the identification of your bacteria. Remember, everyone, you have 1 more minute. Yeah, right there. This is actually good. If you have your slides as well, just make sure that you place them there. I won't put the timing. I don't want to add further suspense. But oh yes. Yeah. Yeah. Okay. I did want to mention this is possible that the bacterium they don't have to be so very so if you're confident with it. Everyone in the meantime, you can start cleaning up and preparing. Really make sure you do based on what you feel and concentrate. At least in school. I would do the both of these. Go with your non-motility if you see it with your motility test tube. If you find that it's indeed the case, you could say you could assume similar for your agonist. Your number was 102. 102. Just one motion, then you could assume it is Brownian. If it's actually moving in a specific direction, one direction.
If you look at what you did, you stabbed outside. Then you went inside, because you still have stabs on that. Yes.
And apart from that, everyone, this is the time to essentially start sending me your slides, because we don't have much time, right? So I'm just going to reiterate this for everyone. You have your lab stain, right on the above, shown with the label, right? Negative, positive, unknown, and positive. Your endospore stain, you're going to submit as well, and your completed data sheet and flowchart. Please make sure that your slides are arranged, aligned as pairs, so that way I'm able to note which one is yours and not have them separated by different individuals. Now is your time. Please make sure that you clean up your bench. You put remember the depression slides, and you go back into the handy drop. Your Wescodine includes your other slides, the capsule stain, endospore stain, and gram stain come here. But your cover slips go into the autoclave. Your used tissue paper, Biblis paper, this is where you also, if they've contaminated by bacterium, you place them into the autoclave. Any other items can be placed back in the respective areas, either in garbage or for exercise 3 down that side, with the petri dishes of 95% ethanol.
I marked my orders here.
You put your sign in, you're ready to go. Once you give me your data,
And I would clean the table.
perfect, you're ready to go. And then you touch right?
Yep.
Perfect. 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 You guys are ready? Perfect. I would recommend if it's at the bottom of the place. You can.
All year.
If you have oil on your slides, just bear in mind that you're able to very gently, not take out any stain, but just gently remove the oil, so that way, like I don't mind there being often makes it difficult for future analysis, so just bear that in mind. Apart from that, everyone, this is the time that we need to clean up. To be safe, you guys keep it as is. You just place them onto the slide box. If of course you have any other ideally it's not the case that you have other liquids, but if you have anything else on your slide, just make sure that you have them dry, essentially. Now, everyone else, please make sure that your flow chart, your data sheet, you've returned them to the front desk. You can also give me your slide or place them into the slide box. When you're done, let me know. No.
Also, for mine, I also put that there's like yellow qualities present. Is that like as a reason?
Everyone,
Make sure you write that down.
please make sure that you shut off your microscopes. And by the way, all stains should be aligned straight. I know it may be tense, Aman. Can you make them all straight for the stains? Crystal violet, none, yeah, perfect. That's okay. If you have your name, don't forget. Perfect. Don't forget to also give me your slides. Make sure they're on the left-hand side. Reason being is because I have another practical, so I want to make sure that they're arranged in the right location. So just bear that in mind, everyone. As long as like you like okay, that's okay.
He said left side, right? Did you say right or left? He said left side, right?
That's yes. In the future, I would say, let's say, you see that this is the line for you, you divide it by that space. Definitely this will be done. So, for every time you mention, like, because it is a bit past time, I would say by the time everyone should be done, you should. Yes. You could also return your other department. Everyone, make sure you shut off your microscopes. I see that there is one on, so just bear in mind that you shut them off before you leave today. And that everything else is clear. No, it's okay. This should be like I'm assuming it's good.
Do you want me to draw things?
But the issue, ideally you could, but this is a conflict, right? Because how can you have pure motility and a negative?
Because of what's the table, it's showing that it should be positive for hanging drop.
I would say go with negative.
For this one.
Because if you actually didn't see hanging drop like being positive, but you find that your motility test is negative, then you need to go with the one that was. I would go with negative. Unless, yeah, like to confirm or validate your result. Here's what I would do.
Is it okay if I send you an email with my post up?
So isn't it if you separated like on you?
Mine is, yes, that's right.
Okay, because it's like there's no room to put everything all in. Okay.
Sure, I'm not sure.
Everyone done? Make sure that you close the lids of your stage. You can use the ground stage, 95% depolyrizing, ethanol.
My old bring some to my house.
If you want to use the ground stage, like, find it, you want to use it better, then
I turned in the one that I did today.
you can find that one better.
Yeah, that. Especially when you got the processing is fine.
Yeah, that's the whole majority is.
Yeah.
You know, the thing is, situation you have.
Someone is
Did you treat it that way?
Yeah, no.
Don't understand your West with that. Stay in. Have your West with stay with. Yeah. You want to see the video? No, I went to that phone for the class right and they the local.
Everyone, the times that we will be cleaning up, so just bear in mind that your microscopes are off. Your tubes, the ones that we use for exercise 3 are referred to the blue right there. Make sure as well that once again, you have your slides labeled. Everything should be organized, so that way other people that are using the lab for later on don't have to worry about re-cleaning up their lens or having any bacteria found on your lens. Integrate that in mind. When you're done, you're ready to go. Have a great day.
And it's not all like you learn from it.
I'm good.
Actually melted, we stuck it on. Not my students, but hey, thanks. Someone asked them. What's up, dude? I tears. Why? I didn't. I have. Little pressure. These make you guys.